It's my pleasure to introduce the meningococcal vaccine session with this work group update. On this slide are the members of the work group, including the ACIP members, the work group leads of Jessica McNeil and Manisha Patel, and the liaison other representatives. You've just heard that one serogroup meningococcal vaccine was licensed just yesterday, the RLP-2086, or Trumemba from Pfizer, for age 10 to 25 years of age. A second meningococcal conjugate vaccine, 4C-MenB, or Bexsero from Novartis, is currently under FDA review for the same age group. As was pointed out, these meningococcal B vaccines are distinct from the conjugate ACWY vaccines because they're based on immunity to proteins rather than capsular polysaccharide. Meninge B vaccine has been used for outbreak response. In 2013, 2 universities experienced outbreak of serogroup B meningococcal disease with a combined 13 cases and 1 death reported. Vaccination campaigns were conducted at both universities in response to the outbreaks using one of the investigational Meninge B vaccine, the Novartis vaccine, obtained through an expanded access investigational new drug, or IND, application sponsored by CDC. In addition, guidance for use of serogroup B vaccines in institutional outbreaks has been developed, and the link is shown on the bottom of the slide. Today's sessions will consist of the following. There'll be presentations on the 4C-MenB Bexsero vaccine from Jim Wiesel from Novartis. There'll be a presentation on the RLP-2086 Trumemba vaccine from Pfizer by Laura York. There'll be a presentation on the epidemiology of serogroup B meningococcal disease in the United States by Jessica McNeil, and followed by an overview of the serogroup B meningococcal vaccines and consideration for use by Manisha Patel. No votes are anticipated at this session. In terms of a timeline for the next ACIP meeting in February 2015, there'll be proposed recommendations for the use of the serogroup B vaccines in persons greater than 10 years of age with high-risk medical conditions, laboratory workers, and outbreaks. The ACIP meningococcal work group will review data on the Meninge B vaccines as it becomes available and continue discussions on use and potential impact of these vaccines. And updated outbreak control guidelines will be developed which address outbreaks due to all serogroups. If I can call on Jim Wiesel.
Thank you for giving me the opportunity today to present data on 4C-MenB, Novartis' investigational vaccine for the prevention of serogroup B disease. Earlier this year, we submitted a BLA with the FDA. I can confirm that the file has been subsequently accepted and that we have been granted priority review status. Today, I want to present the clinical overview for 4C-MenB, and that includes the proposed indication, estimated strain coverage, as well as the vaccine composition. I'll then conclude with a review of the clinical data in the adolescents and adults. That is the age group where Novartis is currently seeking an indication in the US. Based on the proposed indication that's currently under review with the FDA, 4C-MenB will be indicated for prevention of invasive meningococcal disease caused by Neisseria meningitidis serogroup B in individuals between 10 and 25 years of age. The vaccine itself is going to be a sterile suspension, 1/2 ml fill, intended for intramuscular injection. It is fully liquid, so it does not require reconstitution, but it does require refrigeration, cannot be frozen, and needs to be protected from light. Now, the serogroup B capsule is very poorly immunogenic. As a consequence, vaccine strategies have focused on subcapsular protein antigens, but these are highly diverse. To address this diversity, Novartis took 2 approaches. The first was it targeted conserved antigens through a process called reverse vaccinology. The second was that it developed a multi-component approach. That is, by adding multiple subcapsular proteins into the vaccine, the goal was that we would achieve broad coverage against these circulating serogroup B strains. The vaccine is formulated with 50 micrograms each of 3 recombinant proteins: factor H binding protein, Nicero adhesion A, and Nicero heparin binding antigen. The fourth component is 25 micrograms of OMV, or the outer membrane vesicles. Now, this is the same OMV that was used in New Zealand to address a clonal outbreak, so the safety and effectiveness has been demonstrated with over 3 million doses administered in that campaign. All 4 of these components are then adsorbed onto alum. Shown here is the predicted strain coverage for 4-CMNB. Now, coverage is assessed based upon ELISA called the meningococcal antigen typing system, or MATS. Now, the MATS ELISA is correlated with killing of strains by vaccine-induced antibodies in the serum bactericidal assay. An SPA with a titer greater than 4 is considered a correlate of protection against this disease. So in MATS, if a strain exceeds a minimum threshold value for any one of the 3 recombinant protein ELISAs, or its genotype is poor AP1.4, it's highly likely that this vaccine, or this strain rather, would be killed in SPA and is therefore considered to be covered. In the US, we assess coverage based upon a representative panel of 442 strains isolated from individuals between 2000 and 2008. Now, based on MATS, the overall coverage, only 9% of strains were not covered from the strain panel. And of the 91 that were covered, 44% were covered by 1 vaccine component. However, over half of covered strains were covered by 2 or more components in the vaccine. That is, independently, each component was deemed sufficient to kill the strain on its own. The difference between this slide and the previous slide is that this is only for the subset of strains where the disease occurred in individuals between 10 and 25. Now, it's, I think, good to see that the predicted coverage didn't change substantially. It's still 92%. However, what is worthy to note is the number of strains that are covered by multiple antigens did increase up to 61%. So now I'd like to go into a summary of the 4-CMNB trials that were conducted in adolescents and adults. Overall, 18,400 subjects that are adolescents and adults received at least 1 dose of 4-CMNB in clinical studies. In addition, over 6,000 individuals between 2 months and 10 years of age have also received the vaccine, and that contributes to the overall clinical database for this product. But for today, I'll just focus for the remainder of the discussion on those clinical studies that are supportive of the indication we're seeking in the US. 3,139 subjects between the ages of 10 and 50 received at least 1 dose of the vaccine in 6 randomized controlled clinical studies. In addition, over 15,000 subjects received at least 1 dose at Princeton University or University of California, Santa Barbara in 2 CDC-sponsored expanded use IND studies. Those results have been presented to the committee at previous meetings. So today, I just want to focus on 2 studies in particular. V72P10, which is a phase 2b, phase 3 study conducted in Chilean adolescents looking at different dosing schedules as well as persistence of antibodies, and V10203, which is looking at an investigational use vaccine for ABCWY. It looked at different formulations, of an investigational ABCWY vaccine. But what's important in that study was that there was 1 arm where individuals between 10 and 25 in the US received 4-CMNB as a comparator. Now, before I go into that, I just want to give you overall conclusion as to the results of the clinical trials to date. This includes those studies that were conducted at Princeton and the University of California, Santa Barbara, as well as data collected from vaccine post-licensure experience outside the US. 35 countries and over 1 million doses have been distributed. 35 countries have approved the vaccine and over 1 million doses have been distributed. The results show that the vaccine is immunogenic after 2 doses in a variety of schedules. This has also been confirmed in a small US study. And persistence of protective titers have been demonstrated for up to 23 months after the 2-dose schedule. Finally, it shows a demonstrated safety profile. Now, this demonstrated safety profile includes 3 recently conducted large-scale vaccine campaigns. I mentioned 2 previously, Princeton and Santa Barbara. A 3rd was conducted in the Saguenay-Lac-Saint-Jean region of Quebec, where over 43,000 individuals from 2 months to 20 years received at least 1 dose of the vaccine. And what is important there is that the Ministry of Health of Quebec actually did active safety surveillance, including over 12,000 subjects being administered a diary card 7 days after vaccination. And overall, the safety profile seen in these campaigns were consistent with that observed in clinical studies. So now I'd like to go into the V72P10 study. This was a phase 2B, phase 3 randomized observer blind controlled study conducted in Chilean adolescents between 11 and 17, and it evaluated the safety and immunogenicity of various 1, 2, or 3-dose schedules. All 1,625 subjects received either a placebo, 1 dose of 4-CMNB, 2 doses at 1, 2, or at 6 months apart, or various 3-dose schedules. Blood was drawn after each visit, and safety was followed up for 12 months. Here's the results of the primary endpoint of the clinical study. It shows percent of subjects with HSV-A titers greater than 4. Now, immunogenicity was measured for each vaccine component by use of what we called an indicator strain. These indicator strains were uniquely susceptible to killing in SBA to only one component of the vaccine. And now, in the interest of time, I'm not going to show you the results from the 3-dose arm because there was no statistically significant difference between HSV-A geometric mean titers for those who received 2 doses and those that received 3 doses. So the vaccine demonstrated robust immune responses in this age group with several 2-dose schedules. 1 month after the second dose, between 99 to 100% achieved HSV-A titers greater than 4. And as you can see, there was no difference in seroresponse rate across dosing schedules. Now, I do want to mention that for NHBA, we did not have an indicator strain identified at the start of this study, so the analysis was done post hoc. And in addition, because of this, not all of the analyses, as you can see, were conducted. The other thing I want to point out here is that for NHBA, in this study, there were high pre-titers to the strain, presumably due to previous nasopharyngeal carriage. I will show a study later on with much lower pre-titers with immunogenicity as well. Included here are the corresponding HSV-A geometric mean titers for this study, again done at baseline 1 month after 1 dose and 1 month after various 2-dose schedules for each of the 4 vaccine components. Robust responses were indicative for each of the 4 vaccine components and for all 3 2-dose schedules. Tolerability was assessed in this study by comparing against an alum-based placebo. And as you can see here, it's stratified by severity. Local reactions in general, the most common were pain at injection site and erythema, while the most common systemic reactions were malaise, myalgia, and headache. And both were the most common in both the vaccine group and placebo group, respectively. In terms of reactogenicity was higher in recipients of the vaccine compared to the placebo, but the majority of these were reported as mild to moderate and transient. Fever was uncommon and relatively similar in both groups, and fever above 39 degrees was rare. There was no evidence of increasing severity with subsequent doses, and tolerability was acceptable to study participants, as evidenced by the fact that there was low rates of study withdrawals. Persistence of antibodies was measured between 18 and 23 months after the completion of the 2-dose series. It was 18 months after the 0-6-month schedule, 22 months after a 0-2-month schedule, and 23 months after a 0-1-month schedule. And as would be as shown here, you can see the percent of subjects with HSV-A titers greater than 4. And overall, you can see that the majority retained seroprotective levels, with 75 to 95% maintaining titers above 4. And dosing schedule here did not appear to have an impact on the persistence of the seroresponse rates, or seroprotective rates. Antibody persistence shown based on HSV-A GMTs is shown on this slide at 18 to 23 months after the completion of the 2-dose series. As would be expected, you can see GMTs overall did wane. However, they still remained higher than their corresponding pre-vaccination titers. So I'd like to just finish up real quickly with a review of study V10203, which is a study that was done to look at different formulations of ABCWY vaccine in US and Polish subjects between 10 and 25 years of age. Now, I do want to highlight here that in this study, there was one arm where 4-CMNB was used as a comparator, where it was given at a 0-2-month schedule in 10 to 25-year-old US subjects. And this is the arm that I'm going to show you data for in the next 2 slides. So here's percent of subjects with titers greater than 5 for each of the corresponding FHBP and ADA, and NHBA antigens at baseline and 1 month after a 2-dose series. You can see that baseline titers here compared to Chile were much lower. However, robust responses were achieved with 73 to 90% of adolescents achieving titers above 5. And finally, here are the HSBA geometric mean titers for that same study in adolescents between 10 and 25 years of age. So I'd just like to summarize by saying again that BLA for 4-CMNB has been accepted by the FDA. Priority review status has been granted. And overall, based upon the studies, we believe that 4-CMNB generated a protective immune response in adolescents after 2 doses, and this was confirmed in a small US study. Persistence of antibodies was demonstrated up to 2 years after the last dose, and there was no statistically significant difference in HSBA geometric mean titers between subjects receiving 2 doses or 3 doses of vaccines when looking at the indicator strains. We've also begun to collect real-life experience with this vaccine. I said previously the vaccine was approved in 35 countries. There have been over 1 million doses that have been distributed. And from the safety data that has been generated, which includes over 30,000 doses at Princeton and UC Santa Barbara, and over 55,000 doses so far in Saguenay-Lac-Saint-Jean, we can say that overall, no particular concern for any specific event was identified in either campaign. Thank you.
And I think we're going to hold questions and comments until the next presentation. So if we can have Mr. Wassel, or, oh, I'm sorry, Dr. York, thank you.
Thank you. Good morning. I'm Dr. Laura York. I'm the meningococcal lead at Pfizer, and I'm really pleased to be here to talk with you today. Dr. Sun sort of stole my thunder so that I could come in and say we're so excited. We were approved yesterday, and I'm speaking on behalf of many, many people at Pfizer who worked very hard along with FDA to license this product. Trumemba, our meningococcal Group B vaccine, and you've been introduced to it as RLP 2086, and we refer to it very often as bivalent RLP 2086. So I'm going to see if I can advance this. So our vaccine, Pfizer's MenB vaccine, is based on the lipoprotein 2086. This is a surface-exposed factor H binding protein, and you can see from the schematic it's expressed on the surface, accessible to antibodies, and this is expressed in over 97% of invasive meningococcal B isolates. It is a conserved protein, and the gene is rarely absent from MenB. The FHB sequences segregate into 2 genetically and immunologically distinct subfamilies, A and B, as shown in the phylogenetic tree. You can see the subfamily B and the subfamily A, and the length of the line really identifies the similarity between those variants. And you can see that the variants within a subfamily are highly related and have over 84% sequence identity. The bivalent vaccine contains 2 lipidated FHBP variants, an A05 and an B01, one from each of the subfamilies to give that complete protection across the 2 subfamilies. This vaccine design of a bivalent lipidated protein was supported by preclinical data. We looked at monovalent vaccines or a bivalent vaccine composed of a subfamily A and B. This is in rabbits who were immunized with the monovalent or the bivaccine. And you can see that we then looked at the immune sera and the ability of that immune sera to kill diverse MenB strains in an HSBA, and those strains would express either subfamily A or subfamily B as shown along the x-axis. And we looked at the ability to kill in terms of looking at a serum bactericidal assay that included rabbit complement. So if a rabbit is inoculated with a monovalent vaccine of an A05, you can see that there are good responses across the variation in the A subfamily, but not so much across the variants, the strains that express the B variants. Similarly, you can see this predominantly subfamily-specific type of response if inoculating animals with a BO1 monovalent. Good responses across the strains that express those B variants, but not across the A. So very much a subfamily predominant response, and only a vaccine that contains both of these subfamilies provide the protection across the strains that express both either A or B subfamilies. These data really were what supported us moving into clinical trials, and we opened our IND in 2008 and began the discussions with the FDA very collaboratively to define the licensure pathway for MenB vaccines in the U.S. A significant step was the decision that we could use the serum bactericidal assay incorporating human complement as the correlate to predict protection. And in June of 2012, we came to agreement on the immunogenicity endpoints based on the measurement of HSBA responses. And all of our ongoing studies were then modified to provide prospective blind data based on those agreed upon endpoints. That length of time that it took to come to those agreements really, I think, reflects the complexity of defining what the endpoints could be that would then really evaluate and demonstrate a vaccine would be able to protect broadly across the variation that we see in this variant, in this protein, sorry. So these next two slides, I'm going to show you the licensure criteria that was agreed upon with the FDA. Of course, we would have to have sufficient numbers of U.S. subjects to show both safety and the HSBA response. In a clinical trial, there's limitations in the amount of serum you have. To be able to test, you also have to have validated assays with strains. And so you're limited in the number of strains that you'll actually be able to test. However, in phase 1 and 2 clinical studies, we had HSBA data that clearly supported and confirmed the information that we had developed in the preclinical studies that the immune sera, following 3 doses, was able to demonstrate activity against strains bearing different variants. And these are shown by the blue dots on the phylogenetic tree. And so we came to agreement that the HSBA responses against 4 MenB test strains, 1 month post-dose 3, was the endpoint that we could have, so 4 strains. And so 4 strains were selected in an unbiased fashion. These strains would then represent the epidemiologically relevant strains in the US and the diversity of the FHBP being expressed. The FHBP variants expressed are not homologous to the vaccine components. So you can see the vaccine components in the orange dot, the BO1 and the AO5 that I introduced earlier, and the test strains that we have for the licensure criteria are B24 and B44, or express A56 and A22. So they're not homologous to the vaccine components. The endpoints for our studies are, we have 5 endpoints. The proportion of subjects achieving a greater than 4-fold increase in HSBA titer from baseline to post-dose 3 for each of the 4 strains. This really takes into account any background level and defines a clear vaccine benefit in vaccinees. And I do have to point out that the minimum response, therefore, is an HSBA titer of greater than 1 to 16, which is clearly higher than what has been posed as a correlative protection by Goldschneider in the early studies of 1 to 4. And a 5th primary endpoint of the proportion of subjects achieving a post-dose 3 HSB titer of greater than 1 to 8, in fact 1 to 16 in a validated assay of the strain expressing A22, for all 4 test strains, a composite, which is really demonstrating the ability of the antibody elicited by the vaccine in an individual for those antibodies to recognize across the diversity. They recognize the 4 representative strains and therefore will predict activity against the diversity of this antigen. There are also pre-specified thresholds for the lower bound of the 95% confidence interval for each primary endpoint. Again, very stringent endpoints that are going to define that this vaccine will, in all likelihood, be able to provide that broad protection. The clinical development plan for our LP 2086 was really designed for the evaluation and implementation of this vaccine in the adolescent and young adult age group. You can see that there were a number of different studies conducted in the US, Europe, and Australia. In addition to large safety and immunogenicity studies and clinical lot consistency, there were a number of studies that were conducted to look at evaluating the vaccine when given concomitantly with recommended adolescent vaccines. These studies, a number of them are completed and a number are still ongoing. At the time of the outbreak and towards the end of 2013, we had a significant body of data available. And importantly, a number of these studies, the three identified here, a safety and immunogenicity study evaluating concomitant administration of HPV-4, a safety and immunogenicity on a dosing schedule done in the EU, the HPV study done in the US, and the safety and immunogenicity with concomitant with Repavax, a vaccine that's used in adolescents in Europe. These three studies were designed for a different purpose, but in fact, the HSBA data generated in those studies were done using the validated strains and expressing those antigens. And in fact, those protocols had been amended to include those licensure endpoints. So it was possible for us to move forward and submit our biologic license application to FDA for an accelerated approval and priority review, as spoken about this morning by Dr. Sun. We submitted that on June 16th, and as you know, we had approval yesterday on October 29th, and we're all very excited about it. The licensure was based on the clinical data from pivotal trials involving over 4,500 subjects, demonstrating an acceptable safety profile and HSBA responses that met all of these pre-specified endpoints. Because it's an accelerated approval, there are conditions. We do have to provide confirmatory data, verifying the data in these pivotal studies. And those data will be provided from the ongoing studies that you can see are well underway, and those would be submitted post-approval. The indication and usage for Tremembra I've shown you here. It's indicated for active immunization to prevent invasive meningococcal disease caused by Neisseria meningitidis serogroup B. Tremembra is approved for use in individuals 10 through 25 years of age. The approval is based on demonstration of immune responses as measured by serum bactericidal activity against 4 serogroup B strains representative of prevalent strains in the US. The effectiveness of Tremembra against diverse serogroup B strains has not been confirmed. As I said, in terms of the accelerated file, it's administered on a 0 to 6 month schedule. These tables, the next two tables will show you the data from those pivotal studies. Study 1 being a concomitant study with HPV done in the US. Study 2 is that 2 versus 3 dose schedule in the EU, and another EU study, Study 3, looking at concomitant administration of the vaccine with a DTaP IPV containing vaccine, Repavax, that's used, as I said, in adolescents in Europe. You can see that the endpoints, the individuals with 4-fold rise in SBA titer 30 days post-dose 3 on that 0 to 6 schedule, the strains that were used expressing the A22, A56, B24, and B44, and the success criteria of the lower bound are shown in those cells as well, with the lower bound, for example, of A22 having to be greater than 75. The composite endpoint is also shown there, and the lower bound of that is greater than 75%. Again, very rigorous endpoints that will define that this vaccine will be effective. If we focus on the administration of the vaccine, our LP 2086, and the responses, you can see that is a very high proportion of individuals that respond to each of the 4 strains with a 4-fold rise in their HSBA titer, 30 days post-dose 3. And the composite response, again, is very high. Each one of these endpoints meet the lower bound criteria that were set by FDA. Similar results were seen in Study 2, and you can see, again, a high proportion of individuals responding with a 4-fold response to those indicator strains or those predictive strains representative of the strains in the US, and a high response in terms of the composite endpoint. Again, the antibodies being able to recognize across all 4 strains expressing diverse, different FHBP. Administration of this vaccine, Trememba, with HPV-4 does not impact the response. The same similar response rates in terms of fourfold rise and the subjects achieving post-dose 3 titers greater than 1 to 8 for all 4 strains, that composite response. And when given with DTaP IPV, again, an EU licensed product, the 4 endpoints that were looked at, evaluated in this study, met the 4-fold rise and the lower bound criteria for each of the 4 strains. The composite, and you can see across all 4 of these 3 studies, but 4 cohorts, very similar responses. I'm showing here in this table the data that actually make up that composite response, looking at titers of greater than 1 to 8, 1 to 16 on the A22 strain, and the percentage of subjects achieving that titer after receiving the full vaccination series. Again, the same study 1 in the US, study 2 and 3 in the EU, but you can see that the responses are very robust with a high proportion of individuals achieving that greater than 1 to 8 titer after receiving the vaccine to all 4 of those strains. In summary, the HSBA responses to Trememba, the bivalent RLP 2086, elicits SBA responses in completed trials that met all of the pre-specified primary licensure endpoints agreed upon with FDA and indeed with other regulatory agencies. The licensure criteria are designed to demonstrate vaccine-elicited responses that have the potential to provide broad protection against diverse MenB strains. And I think this is really well illustrated in an exploratory analysis we did. Though this is a very small study, it's looking at the ability of the immune sera to elicit HSB responses against serogroup B isolates from recent US university outbreaks. You can see this is a small study, as I said, with just the sera of post-dose 3 and pre-sera in 9 individuals from one of those studies. You can see that there's a significant rise, 4-fold rise. The percent of subjects that achieve that 4-fold rise are high against the isolates from University B that are expressing a B24 variant. That is one of the variants that's in fact, our test strains, so that perhaps it's not unexpected. And the percent response is very similar to what we had seen in the pivotal studies. And the response against those in University A is also similarly very high, percent subjects achieving a 4-fold rise, showing evidence of activity against these strains, which in fact express a very unique FHBP variant, the B153. In terms of evaluation of safety, the safety data in over 4,500 subjects were submitted to FDA. This is across 7 clinical trials. It's demonstrated an acceptable safety profile. The majority of local and systemic events were mild to moderate in severity after each vaccination and were transient. The most common AEs were pain at the injection site, fatigue, headache, muscle pain, and chills. There was no pattern of potentiation with progressive dosing. The vaccine has demonstrated an acceptable profile when administered with HPV-4 or with the Tdap IPV, as I said, which is not a US licensed product. And the rates of SAEs were comparable between the vaccine recipients and controls, 1.7% versus 1.6% in 4 controlled trials. This table shows you the local and adverse reactions of pain, redness, and swelling within 7 days after each vaccination in that US trial of looking at HPV given alone or with RLP 2086 given with HPV or alone. You can see that the rates of adverse events are higher in those receiving the vaccine as compared to saline. The most common was pain, but generally mild to moderate, and you can see that there's no potentiation across the doses. Systemic adverse reactions are shown in this table, again from the same US study. Fever, headache, and fatigue. Again, higher rates of these systemic reactions in those receiving RLP 2086 as compared to those receiving HPV. The fever rates were low, however, and generally mild to moderate. There were no fevers over 40. And you can see that there's no potentiation with subsequent dosing. And importantly, there's no increase when RLP 2086 Trememba is given with HPV. So in summary, Trememba was designed to provide broad protection against MenB. The vaccine is composed of 2 surface-exposed FHBP binding proteins, 1 from each subfamily, A and B. It's a conserved virulence factor that protects the bug from the complement attack, binding FH and downregulating the complement system. Trememba has been evaluated in adolescents and young adults who are at increased risk of meningococcal disease. The HSBA data from pivotal studies met pre-specified endpoints established with the FDA, predicting protection against prevalent meningococcal serogroup B strains causing disease in the U.S. It has a favorable profile when given alone or with concomitant vaccines. So Trememba is designed to provide broad protection against MenB and the full public health potential of this vaccine. An FDA-licensed serogroup B vaccine will only be realized with vaccination strategies that go beyond outbreak control and target prevention in U.S. subjects at increased risk of meningococcal disease. Thank you.
Thank you much. I think we're open for any questions. Dr. Karen?
Thank you. So I have a couple of questions for Dr. York and couple of questions for Dr. York and then a couple of questions for Dr. York and Mr. Wassel. So with respect to the Pfizer vaccine, I was wondering if you have any data on achieving protective levels after a single dose. So for example, in an outbreak situation, if you were to use this vaccine. This one.
So I don't have data on a single dose for you. I can tell you what those data are. It's between 35 and 50% response rate after a single dose. But I can show you these data in the study. It was done in the EU looking at 2 versus 3 dose regimens. You can see in the first column the responses in terms of a titer of greater than 1 to 8 against the 4 strains. And there's a high percentage of subjects achieving that response. And then we looked at different schedules of 0.6 and 0.2, and you can see that the responses are really, quite significant. However, these wouldn't have met the criteria based by the FDA for licensure. So there's a very good response.
Okay, that was actually my second question, was about 2 versus 3 doses. And then the question for both of the presenters is, the sort of longest data that you have on duration of protection, duration of titers, and also do you have any data on concomitant administration with ACYW vaccines?
So we are generating persistence data now. We have studies where subjects were enrolled in new protocols so that we could extend those studies to generate those data. We don't have them at the current time. With the acceleration with the file, of course, you still have to have people actually go through that time so you can look at the persistence of their antibodies. But we are generating those data and they will be available and provided to the work group as soon as they are. And then in terms of, I'm sorry, I've forgotten this. ACYW and Adacel, we have a study that is completing soon. Those data will also be available in the next couple of months and will be provided as well to the work group. So we will have a number of concomitant vaccines, but the MCV4 and Adacel in a study, yes.
Dr. Reingold.
Sorry. Part 2. So for Novartis, we demonstrated persistence for up to 23 months, and I showed that data here to the committee. That's the longest data we have in the 11 to 17-year-old age group. In addition, we did collect data in V7229 in the UK in university students that went up for 11 months as well, and those were similarly robust. In terms of ACWY, we also plan continuing to do a study with concomitant use with ACWY vaccine, but we don't have those results now.
All right, Dr. Reingold.
So for each vaccine, I'm curious if we know anything about the effects on carriage?
In terms of carriage, it's very difficult in a pre-licensure setting to be able to demonstrate this. In many cases, you really need to do immunization of a broad population to interrupt transmission. So that said, we did try and exhibit some demonstration of a carriage reduction in the UK in university students. We gave 1,000 doses to one group and with 4-CMNB and another 1,000 with a control. And while we didn't meet our primary endpoint, which was demonstrating reduction in carriage 1 month after completion of the 2-dose series, when we did follow it up between 3 months and 12 months, we did see statistical reduction overall in carriage of around 26% in A, C, W, Y, and B isolates.
Dr. Riley.
Can I just, since you're asking both companies, I'm sorry, Dr. Potente. We have not actually looked at our vaccine in terms of being able to impact carriage, and I just want to clarify that with vaccinating, the impact on carriage is actually new acquisitions. And so to conduct a clinical trial, as Mr. Wassel said, is very difficult to actually set those up and be able to ascertain those reductions in acquisition of new carriage. It's not getting rid of existing carriage. So we haven't done those studies to look at our vaccine, but have been looking at the possibility.
Okay, Dr. Riley.
My questions were asked by Dr. Karen. Thank you.
Dr. Vasquez.
My question was about carriage.
Ms. Pellegrini.
All right, so as a parent of 2 teenagers, as I know actually a number of us around the table have teenagers, I'm going to issue a plea here. We are talking here about another adolescent vaccine with 3 doses in less than a year, which may or may not be aligned with the recommendations for the other adolescent vaccines. I will say, as I have at previous meetings, we are lucky to get a teenager into the doctor for a well visit once a year. And I think we just have to pay more attention to aligning these schedules to the maximum extent possible. Right now, we know that the meningitis and the HPV completion rates for these series are lousy. They are not where we want or need them to be. And I fear that if we don't really examine this issue thoughtfully, we're going to end up in the exact same place with a MenB vaccine.
Dr. Byington.
Thank you. My question is for the Novartis product. You are targeting several subcapsular proteins, and I wonder if you have any data on cross-protection with other serotypes or serogroups outside of type B?
Yeah. So we do have limited data on protection demonstrated in an SBA assay for non-B serogroups. There was a publication on serogroup X where we were able to cover the clonal outbreaks there. We've also done work in the UK, Germany, France, as well as Brazil and Argentina, where we've looked at some non-B strains, and we do cover them very well, between 30% coverage all the way up to 95 to 100%.
I know you didn't ask me that question, but if I could just follow up and say that FHBP is a protein that's found. It is a meningococcal protein. It's not specific just to MenB. And so we have preclinical experience that shows that, or exploratory analysis that can show that those sera can be active against the serogroups. But this vaccine was really developed to be used for prevention of MenB.
Dr. Baker.
Same question, a little more detail. So both of you said against other capsular serogroups. There is some, could you say which ones, or is that too hard of a question?
Well, the gene is found in all of them, ACYW. We do have some data that shows against CMY. We continue to look at other serogroups as well, and as Mr. Wassel said, there's a number of different studies that have been done looking activity to different serogroups.
So when you remove the capsular antigen, you're stuck with something very similar to MenB, which is that it's a strain-specific demonstration of a response. So we've done testing in W in Argentina, for instance, and we covered exceptionally well. Does that mean that the W circulating in the US, we don't know. We would have to do the same SBA analysis. So the reason we're hesitant to give actual information about what's pertinent in the US environment is, personally, we haven't tested in the US, but For instance, there's an outbreak of W occurring in the UK right now, and we tested and we cover that exceptionally well. So it has to be answered on a specific basis.
The reason for the question is, as you know, there are 13 capsular types, but there's many less that are prevalent in terms of the cause of human disease everywhere in the world, and especially in the U.S. Thank you for your presentations and for your answer.
Dr. Whitley Williams.
Thank you. Pat Whitley Williams from the NMA. Do you have any data on immunocompromised persons, and particularly HIV patients with functional asplenia, or any plans to do those studies?
No, we have no data at this time, though we have looked at the possibility of doing, following up with those studies as well.
So, while we have not looked at HIV patients, we have, as a post-licensure commitment to Europe, have already initiated a study that's ongoing, and enrollment has been completed, correct? In asplenics and complement deficient individuals. So we expect results of that soon.
And Dr. Pickering.
Yeah, I have two questions. One is I couldn't find in here the adverse event, which is the most common in adolescents, and that's syncope, and maybe you have some data on that. Secondly is Dr. Karen, to follow up on her question, we were unpleasantly surprised with quadrivalent vaccine when we thought we would need one dose and then found out we needed two. Can you take the data that you have and do any modeling to give us a better projection or potential projection of how long the antibodies will last after either the 2-dose or the 3-dose vaccines for some prediction of duration of protection?
We have not done any modeling in that context. We've done modeling in terms of looking at the potential impact and estimating times of depletion of that antibody over time, but we haven't done that to predict the actual information, and since we don't have the persistence data at this point, we have not actually done that. And I'm sorry, your first question was syncope. Really, the most common things we saw were pain at the injection site,
So similarly, in the clinical studies, you don't typically see some of these reports, but we've distributed over 1 million doses, over 100,000 we know have been administered, and we have seen a few cases reported in a post-licensure setting of syncope and vasovagal reactions. In terms of modeling, we have 23 months persistence in Chile. We have 11 months in the UK, and so we have been able to do some modeling, which depends upon, of course, the protection against these indicator strains and not all the host of varieties of strains circulating out there. And based upon that, we do estimate between 6 to 8 years persistence on this vaccine, based on caveated heavily based on modeling.
Dr. Ilana Murray? Forgive me.
Aisha.
Thank you.
Thank you for this presentation. My question to both, especially Pfizer. Do you have data about safety and immunogenicity, and for how long the protection will last in children when it is given concomitantly with the routine childhood immunization? Why I'm asking this, because we introduced the quadrivalent meningococcal conjugate vaccine for children and for adults 4 years ago, and since that time, all the cases of meningitis is in children less than 6 months, and it's type B.
So as I said, this vaccine was studied in 10 to 25-year-olds, and we have looked at concomitant administration with the routine immunization, the other routine vaccinations that are given in adolescents, so HPV-4, and we will have data on Adacel and MCV-4 as well very soon.
Dr. Lehr.
I'm a little confused, so I'm just sort of trying to clarify. Pfizer's vaccine has been FDA approved for general use, if I'm understanding correctly. Novartis's has been approved for special situations. Were they both used at Princeton and UCSB? that was only Novartis's. Am I accurate on that?
So Novartis has not been technically approved for use by the FDA. It was done in Princeton and Santa Barbara under a CDC-sponsored expanded access IND. So technically, it was part of a clinical program. And it was for both Princeton and Santa Barbara. It was the Novartis 4-CMNB that was used.
Okay, seeing a lack of additional questions, if we can have, I'm sorry.
No,
The danger of turning right back to the stage.
I was just clarifying, of course, that Trememba was just licensed yesterday for use in the U.S. Yes.
Okay, if we can have Ms. McNeil come on up and present on the epidemiology of serogroup B disease in the U.S.
Good morning. Today I will be presenting an overview of the epidemiology of serogroup B meningococcal disease in the United States. I will begin by describing the current epidemiology and burden of serogroup B meningococcal disease with a focus on adolescents and young adults and college students, followed by a brief review of groups at high risk for serogroup B meningococcal disease. In the United States, we have two sources of surveillance data for meningococcal disease. The first is the Active Bacterial Core Surveillance System, or ABCs. ABCs is an active laboratory and population-based surveillance system that collects data on culture-confirmed cases of meningococcal disease in 10 states. Cases in the ABC sites can be projected to the U.S. population to estimate incidence. The National Notifiable Diseases Surveillance System, or NNDSS, is a passive surveillance system that all states and territories report data to for all nationally notifiable diseases. NNDSS captures information on cases, including cases confirmed by PCR. However, serogroup and outcome information has historically been limited. Since 2005, serogroup and outcome information in NNDSS has been supplemented by data from state health departments and ABCs to improve data quality. In the United States, the incidence of meningococcal disease is currently at a historic low. In 2013, 564 cases were reported to NNDSS among persons of all ages. Declines in incidence have been observed for all of the serogroups, including serogroup B, which is not included in the quadrivalent meningococcal conjugate vaccines. In addition, despite steady increases in coverage with quadrivalent vaccine among adolescents, much of the declines in incidence occurred prior to high levels of coverage in adolescents. The incidence of meningococcal disease is highest among children less than 1 year of age, and in that age group, 67% of disease is caused by serogroup B. There is a smaller peak in disease incidence among adolescents and young adults, and in that age group, approximately 40% of disease is caused by serogroup B. This figure shows incidence of meningococcal disease by serogroup for adolescents during two time periods. The dashed lines represent incidence of serogroup B and serogroups C and Y combined during 2005 to 2007, and the solid lines represent incidence during 2010 and to 2012. What you can see here is that incidence has declined for both serogroup B and serogroups C and Y combined in the more recent years, but also that serogroup B disease now causes about the same amount of disease as serogroup C and Y in this age group. This change in the proportion of serogroup C and Y disease among adolescents is likely the result of quadrivalent vaccine use in this age group, as this has not been observed in age groups not recommended routinely for vaccination. The estimated annual cases from serogroup B and serogroups C and Y meningococcal disease are shown in this table. We estimate that serogroup B cases in 11 to 24-year-olds have declined from 161 per year in the last high incidence years to approximately 48 to 56 cases annually in the recent low incidence years. The case fatality ratio from serogroup B is 12.5%, which is slightly lower than for serogroups C and Y combined, where the case fatality ratio is 16.6%. Among adolescents and young adults, the case fatality ratio from serogroup B is roughly half of that observed for serogroups C and Y combined. Information on college attendance from meningococcal cases is collected through ABCs but is not collected in NNDSS. During 1999 through 2012, 29% of serogroup B cases in all 18 to 23-year-olds reported through ABCs attended college. We can apply that proportion to serogroup B cases reported through NNDSS in this age group to estimate the number of cases of meningococcal disease occurring in college students nationwide. In the United States, in 2012, approximately 61% of young adults completed high school and enrolled in college. Therefore, we estimate that there are approximately 16.6 million college students aged 18 to 23 years in the U.S. In 2008 to 2012, there were approximately 11 cases of serogroup B meningococcal disease in college students annually, with 1 death in this age group. This is out of 37 cases and 2 deaths among all 18 to 23-year-olds. The incidence of serogroup B disease in college students during the most recent years of 2008 to 2012 was 0.01 per 100,000, compared to an incidence of 0.03 per 100,000 among all 18 to 23-year-olds. While the incidence and number of cases of serogroup B meningococcal disease among college students has continued to decline, there have been 5 serogroup B clusters or outbreaks on college campuses in the last 5 years. These outbreaks have been described to ACIP previously, but have had between 2 and 13 cases per outbreak and have lasted from a few days to nearly 2 years. Fortunately, meningococcal outbreaks remain rare in the United States. However, each of these outbreaks cause significant anxiety and are devastating to the communities in which they occur. To summarize, with widespread use of conjugate vaccine in adolescents and young adults, serogroup B now causes 40% of meningococcal disease cases in this age group. Approximately 50 cases occur among 11 to 24-year-olds, with 1/3 of cases among 18 to 23-year-olds occurring in college students. While most serogroup B cases are sporadic, even in college students, recent outbreaks on college campuses have been due to serogroup B. I will now shift gears and discuss groups at high risk for meningococcal disease. There are 3 main groups of individuals who are considered at high risk. The first group includes those who have medical conditions that place them at increased risk, including those with persistent complement component deficiencies and functional or anatomic asplenia. In addition, microbiologists and the at-risk populations in outbreaks are also at higher risk. While these groups are all currently recommended for vaccination with quadrivalent vaccines, they remain at risk for serogroup B meningococcal disease. Persistent or genetic deficiencies in the common complement pathway are well known to increase risk for meningococcal disease. These deficiencies are rare and only affect about 0.03% of the U.S. population. Individuals with persistent complement component deficiencies are at up to 10,000-fold increased risk for developing disease and often develop recurrent infections. Solaris treatment has not been previously included in the high-risk vaccination recommendations for meningococcal disease, but functionally creates a complement deficiency by binding to C5 and inhibiting the terminal portion of the complement cascade. Several individuals in the clinical trials for Solaris have reportedly developed meningococcal infections despite prior vaccination with quadrivalent vaccine. Persons with functional or anatomic asplenia also appear to be at increased risk for meningococcal disease. However, the data are less compelling than their risk for pneumococcal disease. This group includes persons with sickle cell disease, which affects about 90 to 100,000 persons of all age in the U.S., and in asplenic persons, the case fatality ratio for meningococcal disease is elevated. Microbiologists who work with Neisseria meningitidis are also at increased risk for disease. A high case fatality ratio has been observed among microbiologists who have developed meningococcal infections, likely because of increased exposure to high concentrations of organisms in highly virulent strains. The majority of cases among microbiologists have occurred in people who were not using respiratory protection at the time of exposure, and these were not limited to only research microbiologists who routinely worked with meningococcal isolates. There are an estimated 100,000 clinical microbiologists and about 400 research microbiologists who routinely work with Neisseria meningitidis in the U.S. The final high-risk group includes people in the at-risk group during a meningococcal outbreak. Fortunately, meningococcal outbreaks are rare and only cause about 2 to 3% of U.S. cases. However, as an example of the increased risk for meningococcal disease in the recent serogroup B outbreaks on college campuses, at Princeton University there was a 1,400-fold increased risk for meningococcal disease, and roughly 7,500 people were recommended for vaccination with serogroup B vaccines in that outbreak. In the recently published guidance for serogroup B outbreaks in institutional settings, the threshold for vaccination has been defined as 2 cases in institutions less than 5,000 persons and 3 cases in institutions with more than 5,000 people. This table summarizes the risk groups that we've just gone through and also includes available data on cases that have occurred in each of these groups. Excluding the at-risk population in outbreaks, we estimate that there are less than 300,000 individuals who fall into these high-risk groups. Although only a handful of cases have been documented in ABCs or the published literature, these groups are known to be at high risk for meningococcal infection and are currently recommended to be vaccinated with meningococcal conjugate vaccines, but remain at risk for serogroup B disease. In summary, the incidence of meningococcal infections is declining, including for serogroup B. In recent low-incidence years, approximately 50 cases of serogroup B meningococcal disease occurred in adolescents and young adults each year. In addition, persons in high-risk groups who are recommended for vaccination with quadrivalent meningococcal vaccines remain at risk for serogroup B meningococcal disease. Thank you.
Thank you much. And I think we are going to move to Dr. Patel's presentation and then open up for questions thereafter.
Good morning. So the meningococcal work group has been discussing serogroup B meningococcal or MEN-B vaccines on work group calls. So for this talk, I'll be providing a summary of some key points regarding these vaccines and the work group's considerations for use of MEN-B vaccines in the U.S. Development of MEN-B vaccines has been challenging. The polysaccharide capsule is the vaccine target for MenACWY vaccines, but is poorly immunogenic for MEN-B because of antigenic similarity to human neuronal cells. MEN-B vaccines that target other antigens, such as outer membrane vesicles, have been used to control several MEN-B outbreaks in the past, including a large epidemic in New Zealand in the early 2000s. However, these vaccines are strain-specific and provide limited cross-protection with heterologous strains, have a limited duration of protection, and have limited efficacy in younger children. Ideally, vaccine targets would require antigens to be an essential gene, be immunogenic, have relatively low diversity, and be surface exposed. Recently, several outer membrane proteins that are found across all meningococcal strains, regardless of serogroup, have been identified that generally meet these criteria. Factor H binding protein, or FHBP, allows the bacteria to evade complement-mediated lysis. FHBP is classified as either subfamily A or variant 2-3, or subfamily B or variant 1. Neisseryl heparin binding antigen, or NHBA, promotes bacterial survival in the blood, and Neisseryl adhesin A, or NADA, allows the bacteria to adhere to and invade epithelial cells. Although these proteins can be found on the majority of MEN-B strains, they are antigenically diverse and can therefore impact vaccine effectiveness. There are 2 MEN-B vaccines for persons 10 to 25 years of age being considered in the U.S. As mentioned in the earlier presentations, RLP 2086 is manufactured by Pfizer and is a 3-dose series administered at 0, 2, and 6 months. It contains both subfamilies of FHBP and was just licensed by FDA yesterday as Trumemba. 4C MEN-B, or Bexsero, which is manufactured by Novartis, is a 2-dose series administered at 0 and then 1 to 6 months and contains FHBP subfamily B variant 1, NHBA, NADA, and port A1.4, which is the same port A type as the OMV vaccine used to control the New Zealand epidemic. It was licensed in Europe, Australia, and Canada in 2013 for persons greater than or equal to 2 months of age, and it was administered to almost 17,000 participants under an expanded access investigational new drug protocol to control 2 recent university outbreaks in the US. And in light of these outbreaks, it was granted a priority review designation with an announcement regarding licensure expected by early 2015. Because of the low incidence of meningococcal disease in the US, pre-licensure trials using clinical outcomes would require very large sample sizes and thus are not feasible. Human serum bactericidal activity, or HSBA, has been shown to correlate with protection against meningococcal disease and is based on studies conducted in the 1960s that demonstrated bactericidal antibodies were protective against invasive serogroup C disease in military recruits. Bactericidal antibodies in persons immunized with OMV vaccines during various MEN-B outbreaks also correlated with protection. Based on these data, HSBA was established as the serologic marker to infer protection with MenB protein vaccines during the 2011 VRBPAC meeting. However, there are several challenges in assessing immunogenicity of MenB vaccines. First, evaluating clinical efficacy in higher incidence countries would not be appropriate for licensure in the US because the molecular epidemiology differs between countries. Second, the measurement of HSBA is assay specific and requires validation each time a parameter, such as strain or antigen, is changed. And because of the antigenic diversity among MenB strains, bactericidal activity against multiple strains is needed to evaluate antigen-specific responses and address cross-protection with heterologous strains. However, the number of strains that can be tested is limited by HSBA methodology, which would require large volumes of sera and identification of a complement source for each strain tested. Because the vaccine targets and the number of vaccine targets differ between the two vaccines, selection of the 4 primary strains to assess immunogenicity was performed differently for each vaccine. For 4C-MenB, strains were selected to evaluate immunogenicity to each of the 4 antigens individually using pooled human sera. For RLP 2086, strains were selected or systematically selected to assess immunogenicity using individual human sera from a representative collection of circulating strains in the US. The differences in the strains selected and the method in which they were selected for each respective vaccine is important to consider when interpreting immunogenicity data. A second key difference in how immunogenicity was assessed for the 2 vaccines is the primary endpoints. These primary endpoints were presented earlier by the manufacturers and are restated here. In summary, for 4C-MenB, 73 to 100% of adolescents demonstrated protective titers, with titers decreasing by 5 to 25% at 2 years post-vaccination, depending on the antigen that was tested and the population evaluated. For RLP 2086, 75 to 100% of adolescents demonstrated protective titers following 3 doses, depending on the primary strain tested, the study population, and the endpoint that was used. Recognizing that the primary endpoints differ for each vaccine is also important to consider when interpreting immunogenicity data and comparing vaccines. The polysaccharide capsule is highly conserved among strains within each serogroup, and therefore vaccines that target the capsule, such as quadrivalent meningococcal vaccines, typically do not require assessment of breadth of coverage. Because the vaccine targets for MenB vaccines are antigenically diverse within circulating MenB strains in the US, a multifactorial approach can be helpful in estimating coverage. This includes determining the presence or absence of the gene, genetic sequences, level of expression of the antigen, and bactericidal activity by HSBA. In this study, the first 2 factors were assessed, presence or absence of the gene and the genetic sequences. 650 MenB isolates were collected through ABCs from 2000 to 2008 and then sequenced for the 3 different vaccine targets, FHBP, NADA, and NHBP. And the purpose of this slide is just to show you the degree of genetic diversity in the subvariants circulating in the US, particularly for FHBP and NHBA. Genetic sequencing showed that all MenB isolates contained FHBP, and that 59% were subfamily B, or variant 1, which is contained in both vaccines, and 41% is subfamily A, or variant 2-3, which is contained only in RLP 2086. NAD-A, which is included in 4C-MenB, was present in only 39% of all MenB isolates. Because NAD-A is found in less than 10% of isolates that were FHBP subfamily A or variant 2-3, which is not included in the vaccine, based on this analysis, NAD-A would only increase coverage for 4C-MenB from 59 to 63%. NHBA is highly diverse and found on all MenB isolates, but as presented earlier, elicit lower titers of bactericidal antibodies. And then POR-A 1.4, which is also included in 4C-MenB, was found in less than 5% of MenB isolates. Assessing breadth of coverage can be complicated and was performed differently for each vaccine. For RLP 2086, FHB sequence analysis and measurement of surface expression by flow cytometry was performed for an epidemiologically representative collection of over 1,200 MenB strains, of which 432 were collected from ABC surveillance sites. Their analysis demonstrated variability between subfamilies as well as surface expression of FHBP. In a subset of isolates for which bactericidal activity was assessed, isolates with moderate or high-level expression of FHBP was predicted of bactericidal activity, whereas there was lower correlation among isolates with low expression of FHBP. Novartis used a different approach to assess breadth of coverage of 4C-MenB. Because there are multiple antigens contained in the vaccine, there are limitations on how many strains can be tested by HSBA for the reasons I mentioned previously. The meningococcal antigen typing system is a sandwich ELISA that was developed to measure both cross-reactivity with vaccine antigens, as well as the level of expression of each antigen to predict bactericidal activity against a broad number of strains. MATS was previously bridged to HSBA in a subset of antigenically diverse strains and was found to be greater than 80% predictive of bactericidal activity when one antigen was expressed above a certain threshold, and greater than 90% when at least two antigens were expressed above a certain threshold. Based on their analysis of over 400 isolates collected through ABCs, 4C-MenB is estimated to cover 91% of circulating strains in the US. Differences in how breadth of coverage was estimated by the manufacturers is important in understanding how well each vaccine will protect against endemic disease. So in summary, there are two MenB vaccines available in the US, RLP 2086, which was recently licensed as Trumemba, and 4C-MenB, which is currently undergoing priority review by FDA. A serologic marker was used to infer protection against MenB disease. Demonstration of breadth of coverage against diverse strains will be critical for evaluating vaccine effectiveness. And last, there could be potential differences in immunogenicity and breadth of coverage between the two vaccines due to their different vaccine targets. So I will now discuss the workgroup considerations for use of MenB vaccines in the US. As Jessica presented earlier, rates of meningococcal disease have reached historic lows and are decreasing for all serogroups. Vaccination with MenACWY conjugate vaccine is recommended for adolescents 11 to 12 years of age, with the booster at 16 years of age. Increasing coverage with these vaccines has contributed to decreasing rates of disease among adolescents. However, MenB disease continues to contribute to about 40% of meningococcal cases, with 50 cases reported annually among adolescents in recent years. There are a number of challenges to keep in mind when considering use of MenB vaccines in the US, and I actually think a lot of you had brought this up in the earlier discussions. So first, breadth of coverage has only been estimated. Actual breadth of coverage is unknown. Although we have a little data for 4C-MenB, duration of protection and impact on carriage is unknown. The potential impact of vaccine pressure on circulating strains will be unknown until these vaccines are used more broadly. The multi-dose schedules required for both vaccines make implementation challenging. And finally, it is important again to note that the burden of MenB disease is low, and not all cases will be prevented by vaccination. The work group has been discussing a number of different options for use of MenB vaccines. To address the immediate need of protecting high-risk groups, the work group is considering vaccination of persons with medical conditions high risk for meningococcal disease, such as persistent complement component deficiencies or anatomic or functional asplenia, microbiologists, and use in outbreak response. The work group has also been reviewing available data and discussing options for routine recommendations for expanded groups. Although the vaccines were reviewed or are currently undergoing review through an accelerated pathway, there is still additional data needed for both vaccines to inform policy decisions. This includes duration of protection, immunogenicity against additional strains to evaluate breadth of coverage, safety and immunogenicity of concomitant vaccination, high-risk groups, as well as other age groups, and additional safety data. A number of these studies are ongoing and will be reviewed by the work group once these data are available. As I mentioned, the work group has prioritized considerations for vaccination of high-risk groups in persons greater than or equal to 10 years of age. As Jessica presented earlier, persons with high-risk medical conditions and microbiologists account for less than 300,000 people in the US. And based on the interim guidelines for use of MenB vaccines in institutional settings, vaccination would have been recommended for all 5 MenB outbreaks reported on college campuses over the past 5 years, approximately 60,000 people. The overall goal will be to align the high risk and outbreak recommendations for both MenB and MenACWY vaccines. Thus, the current plan for the February ACIP meeting is to present grade for high-risk groups and propose language for use of MenB vaccines in persons greater than or equal to 10 years of age with high-risk medical conditions, microbiologists, and outbreaks for a potential vote. Additionally, for the June and October meetings, we will review the evidence for expanded target groups, which includes grade and economic and impact analyses, and present the updated outbreak guidelines for all serogroups. In conclusion, considerations regarding use of MenB vaccines in the US will be complex. Additional data following licensure will help inform policy decisions, and the work group will continue to review data on MenB vaccines as it becomes available and engage in discussions on use of these vaccines for expanded groups. So we were hoping to get feedback on this two-tiered approach, as well as any additional data the committee would like presented at future meetings. And that's it. Thank you.
I want to thank both Ms. McNeil and Dr. Patel for just very nice, well-organized, succinct talks. These were just wonderful. I open it up to any questions from the committee or liaisons. And Dr. Harrison.
So Bexsero is licensed elsewhere starting at 2 months of age, and I was wondering what the plans were for licensure in the U.S. Down to that age range.
So in terms of licensure of Bexsero at 2 months, we have decided that we made progress enough on an ABCWY vaccine, and I've shown previously the results phase 2 different formulations. So I think we are trying to go forward with an ABCWY vaccine in infants in the US, but that's several years away.
Other questions? Dr. Baker.
I just have a question on the epidemiology, and it's not relevant to the age group that you're targeting. So if you look at college students, the current incidence is 0.01 per 100,000, yet in infants less than 1 year of age, and this is serogroup B specific, it's 1.5 per 100,000. Do you have any data within that less than 1 for the first 6 months? And has anybody thought, just to raise, it's a rhetorical question, about maternal immunization?
So we do have data in children less than 6 months, and that was presented several ACIP meetings ago, but a lot of the disease actually is in the younger infants. And I think your question about maternal immunization is a great one.
Well, and the question about maternal is, if we say high risk, if a microbiologist is pregnant, would you know, I would assume that she would be vaccinated. Another question is, what about travelers to serogroup B in hyperendemic or epidemic areas?
Yeah, we haven't really considered that at this point, but it's something to discuss.
Dr. Belancha.
So just a question, what are the considerations for potential use in microbiologists who presumably are, many of which are over age 25, given that the license recommendation now is 10 to 25?
So these sort of the granularity of the microbiologist recommendations we're going to be discussing within the work group. But at this point, what we're suggesting, it would be individuals greater than or equal to 10 years of age. The difference is as you get younger, the doses change, the immunogenicity is different, but we expect that the immunogenicity would be similar for the older age groups. But this is, again, something we'll continue to discuss within the work groups.
Dr. Byington.
Is the work group discussing for college students a routine type of recommendation or only consideration in outbreak settings? I was not clear on that.
So the outbreak, the sort of high-risk recommendations that we're considering for February would, that's the outbreak response. The routine recommendations, college students, adolescents, we are discussing that. Like I mentioned, there's a lot of data that we're still waiting to receive. We're actually reaching out to the non-ABC sites, the meningenet sites, for instance, to get a better understanding of college cases within other states that are just not ABC. So a lot of data that we're still going to be reviewing.
I'm curious at this point, now that there is a newly licensed vaccine here and Bexsero has been used, do we have a price on those two vaccines for the U.S. market?
Good morning, Angela Wong, representing Pfizer Vaccines in the U.S. Our priority currently has been about securing supply, which we expect shortly, and our decisions around pricing because of the rapid nature by which this approval has taken place is still being finalized. So as of today, I do not have a price for you, but we will have pricing shortly, commensurate with the supply that we are advancing just as rapidly as possible.
Thank you. And
Clem Lewin, Novartis Vaccines, as our vaccine is not licensed yet, as we all know, we do not discuss pricing prior to licensure.
Thank you. Yes, Dr. Even.
I think because the vaccine is available now, the first question I'm going to get in the college health world is when is it available? I mean, when will the vaccine actually be available? Because our age group is included in the licensure.
So we are working as quickly as possible to advance the supply. We expect within the next 2 to 3 weeks for this to be available for Pfizer.
Up at the microphone, please.
Laura York with Pfizer. I just wanted to, again, emphasize that Trumemba was licensed to prevent MenB disease. And the discussion of the coverage, the breadth of coverage, I think the phylogenetic tree and the activity against those, the diversity of the FHBP really addresses coverage and the fact that this is a protein which is conserved and present in all MenB is going to be used to protect that bug. Its function is to protect it against the immune system and to evade it. The other thing is that, in fact, we've moved very fast to license this vaccine, and I know the work group is working very hard to address this. And I'm just wondering if there's, because the vaccine was developed to actually prevent outbreaks, not to say it's not useful to use in control, but would the ACIP like to see data on the use of this to prevent outbreaks in universities brought forward sooner because of this acceleration of having a vaccine to speak about? And what information would you need for that? Persistence data I can understand in terms of routine recommendation age-based, but do you need those data to move this forward in that risk group?
Okay, gentleman at the microphone.
Rino Rapoli, Novartis. I have a comment about the epidemiology which has been shown. If I understand correctly, you've shown data up to 2013, where basically in the age group we are talking about is there are 50 cases of meningococcus B with 12.5% mortality. Now, those data are perfect, I think, up to 2013. But if I look at what happened during the last year, we got 3 deaths in that age group, one at Drexel, one at Georgetown, and one in San Diego. With the 12, basically that means that with the mortality of 12.5, you will need 300 cases more or less per year to justify that mortality. So I'm not challenging the data before 2013, but clearly in 2014, things seem to be different because there are more deaths than predicted by the epidemiology. So that's an observation. The other thing is that none of this, you're proposing basically to look at high risk and outbreak control at the beginning, but none of these 3 deaths were going to be prevented by just recommending outbreak control. So I wonder whether, we should reconsider that.
Any? Okay, thank you. Other comment at this point in time? At this point, I'd like to call Lynn Bozoff, Stephen and Beverly Ross, and Frankie Miley to the microphone. If you're here in the audience, we have some time for public comment on this vaccine program. And per usual, please state your name, your association, any conflicts of interest, and we try and hold our comments to about 3 minutes. Thank you.
Hey, I'm Lynn Bozoff. I'm president of the National Meningitis Association, and I have no conflicts of interest. Many of you are familiar with my son's story. He died in 1998 from meningococcal disease. There was a polysaccharide vaccine available, but unfortunately there were no recommendations, so my husband and I did not know to have our son vaccinated. In October of 2000, ACIP voted to recommend that college students be educated that they were at increased risk for meningococcal disease and that there was an available vaccine. It was the right thing to do to protect our children. In 2005, the conjugate vaccine was recommended to prevent this disease in adolescents and teens. Again, it was the right thing to do. Later on, the booster dose was recommended to ensure that these kids maintained their protection. It was the right thing to do. I have heard over and over from parents whose children have suffered from serogroup B disease, especially this past year. They all said the same thing. I thought my children were protected. They all wanted the broadest protection possible. I was really thrilled yesterday to hear that the FDA had approved licensure of one of the vaccines, and I look forward to the other vaccine being licensed soon. We now have the tools to protect our children against all 5 serogroups. We need to do the right thing and routinely recommend vaccination. It's the right thing to do. It's what parents do. It's what society should do. We need to protect our children. I also wanted to alert you to a new activity that NMA is conducting. We are going to convene a roundtable that includes a cross-section of meningococcal disease advocates, including survivors, family members, college educators, infectious disease specialists, and any others who have a unique perspective and knowledge about meningococcal disease. Our goal is to prevent the real-life impact of this disease and to give voice to the concerns and viewpoints of those who have been affected. And we hope to have a report to share with the ACIP before the February meeting. Thank you.
Thank you very much. And then Stephen and Beverly Ross.
My name is Stephen Ross, and this is my wife Beverly. We're here today with the National Meningitis Association, and we have no conflicts. We're the parents of Stephanie Ross, and I hope her name still rings a bell with you. She was a Drexel University student who died after contracting serogroup B meningitis in March. She went to bed early one Sunday night because she was feeling tired, and only a few hours later, she was fighting for her life. It was a battle she was destined to lose. She was just 19 years old. She was an intelligent young woman who was attending Drexel University on a Ben Franklin Fellowship. She was well liked at Drexel, as shown by the busload of her students who traveled across the vast state of Pennsylvania the weekend before finals to say goodbye to her. She also went out of her way to assist anyone who asked for help, and even some who didn't. She was quickly growing into a leadership role with her FAMU sorority sisters. When talking about her, we always like to share a quote that was posted on one of the websites that she created before passing in connection with a fundraiser for the Children's Hospital of Philadelphia. It said, "Thank you for always living your life the way it was supposed to be lived, full of love, laughter, and kindness, to not just friends, but strangers as well." Since Stephanie's passing, at least 2 more college students have died from the serogroup B disease. When will it stop? When we need to say enough is enough. As a percentage of all students that populate our country's college campuses, we realize that these deaths represent a small number, but when the student is one of yours, any number above 0 is unacceptable, especially if it could have been prevented. Parents have lots of things to think about when they send their children off to college. We shouldn't have to worry about this disease. Let's do the right thing to help protect our children from this devastating bacteria. We seem to have the means to stop the spread of this disease, as evidenced by the fact that no additional cases have been reported from the Princeton campus since the use of the serogroup B vaccination there. We don't want any more parents to receive a call like we did when you hear that your child has died from contracting the serogroup B meningitis. Now that the FDA has licensed the first of the serogroup B meningococcal vaccines, we hope that you will recommend them for adolescents and young adults. At the very least, they should be recommended for college students, as they seem to be the largest group at risk. Like us, many parents and students have mistakenly believed that previously licensed vaccines protected them against all the serogroups that can affect our children. In the coming weeks, several groups associated with Drexel University will be submitting letters supporting a recommendation for college-age adults. Some of them will be from Stephanie's classmates, who would like nothing more than to be protected from the bacteria that took the life of their dear friend. They want to be able to win the battle over the disease that Stephanie could not conquer. We are here on their behalf, and on behalf of her sister, Jacqueline, who now is an only child and a freshman at one of the largest universities in the United States. It would be a tragedy, like Stephanie's death, to see these vaccines approved by the FDA, but not widely recommended. Thank you for the opportunity to address you.
And thank you for sharing the story of your loss with the committee. And Frankie Miley.
Hi, I'm Frankie Milley, and I'm the National Executive Director of Meningitis Angels. You've shared stats today, and I want to share just a quick stat with you. My mother's twin brother died from diphtheria. Her father died from tetanus. Her cousin died from influenza and the big outbreak in the early 1900s. As I grew, one of my best friends had polio and was in an iron lung. And later he did survive. He was in a wheelchair and very crippled for the rest of his life, which was very short, I might add. My sister had chickenpox and was in the hospital for 3 weeks. We almost lost her. She had serious secondary infections, pneumonia from that disease. My mother died from Hib pneumonia just a few years ago. As you know, Ryan died from meningococcal meningitis. He went from perfect health to blood coming from every orifice of his body in less than 14 hours. My only child, I watched him die needlessly. And I don't share this with a lot of people, but I've been married to my current husband for almost 41 years. But before that, I was married for a year to someone just to show my mother I could. And unfortunately, out of that, I blew a scholarship to college. And I said I came away with half the alphabet. I had TMJ from being slapped around, and I had HPV, which ultimately led to a hysterectomy, which took the part of my body that carried Ryan. It was like losing him all over. We now have Ebola. We don't have a vaccination. I want to commend these people in this room and everybody at this CDC who has worked so hard against this disease to protect us all. You're doing a great job, and keep it up. And I know you don't hear that, but I'm here to tell you you do. If you look at meningococcal disease and you compare it to Ebola, it's really similar. Only sometimes meningococcal disease works much faster, and when they survive, they're left devastated. As you move forward, there's some things I want you to consider. We just, in our organization, we have hundreds of families across the country. We just took in a new family. Their 17-year-old son died in March of this year from meningococcal B. I talked to Scott yesterday, and he was on the way to Canada today to take his other son to get vaccinated against this disease. He can't go through this again. Many parents don't have that opportunity to go to another country and get their kids immunized. So we need to be protective of that. I also want you to consider, we had an outbreak in 2000 in our school district of 6,500. We had 15 cases of meningococcal disease. When I called the health department at the state and told them, asked them if they knew about it, they didn't, and they hadn't had any reports. They came down and we immunized 45,000 people in 3 days in that outbreak. The problem was when I went to the hospital to find out why they weren't reporting, they said it costs money to send the specimens and we're just not gonna do it, even though it's required. So I want you to consider that the case rate may be higher than we think it is, because a lot of times when the kids are brought to the ER, they have symptoms, they're bombarded with antibiotics. We know the bacteria dies. Ryan's cause of death on his autopsy was Frederickson-Waterhausen syndrome, which is a bilateral bleed of the adrenal glands. We know that later that the sepsis from the meningococcal caused that. But I'm afraid that we're missing the actual rate of this disease in this country because we are good at our jobs or because we're not good at our jobs. Either way, that can go either way. Finally, I just think that in a country as great as ours, and the work that you've all done and the work that industry does to bring these vaccines to us, it takes years of research, development, money. Some people research and never live long enough to see their research come to pass. We can prevent this disease. We can't prevent Ebola right now, but we can prevent this disease. Nobody in this country should die from a vaccine-preventable disease. So moving forward, and I know you've got a lot of work to do with this. I know there's still a lot of unanswered questions, but I have faith in this committee right here that at the end of the day, you're going to do the right thing and recommend this for age 15 through 25. And please, I beg you, let's don't go back to that day when we only vaccinate those kids living in dorms. Meningitis doesn't stop at the dorm room door. Thank you so much for your work and your kindness.
Thank you, Frankie, for your comment. Dr. Plotkin.
So I have absolutely no conflict of interest with respect to Pfizer or Novartis. Just want to make that clear. To one specific point and one general point. The specific point is, as was said, 29% of disease in adolescence occurs in college students. Obviously, that means 71% occurs in non-college students who are smoking and going to bars, etc. So that a recommendation for college students is, in a sense, discriminatory. The more general point is one that is obvious, or sort of a 400-pound gorilla, but I want to bring it out publicly. And that is that two companies have spent millions of dollars to develop vaccines against what might be called a minor disease in terms of incidence. And if these vaccines are not recommended, then this puts a pall on vaccine development for diseases that are less prevalent than the ones for which we have general recommendations. And I hope you will keep that in mind for for the future, because this has great implications for future vaccine development.
Thank you, Dr. Plotkin. Dr. Baker.
Just to add to that, what vaccine preventable disease in the United States causes a 10% mortality? And I would second the comment about age group as opposed to college. And then finally, something that I struggle with, I certainly struggled it as chair of the meningococcal work group when the quadrivalent was recommended, as chair of the ACIP, and I still struggle with it, and I don't have an answer. And I know you're in the hot seat in February, it sounds like, but it's very difficult to know how much money we should spend for prevention of rare diseases. And I think that this will increasingly trouble the CDC, ACIP members, the AAP if it's a childhood disease, the ACP if it's an adult disease. It's very, very difficult given the past history. It's not so long ago, really, a little more than a decade, when vaccines were cheap for the prevention of childhood diseases. So it's just a comment.
Thank you. Dr. Offit.
Thanks, John. You know, I felt like I've lived through this once before. When I was on the ACIP and head of the polio vaccine working group in the late 1990s, we made a decision to move from the oral polio vaccine to the inactivated vaccine. We did it because we knew that every year, although we'd eliminated wild-type polio from this country, we hadn't eliminated vaccine-associated paralytic polio. Now, to do that, to move from OPV to IPV, cost roughly $4 million per case of VAP prevented. But we did it because we know we could, and because we believe we could afford it. I think in some ways the analogy is here. It was 6 to 8 cases there. It's roughly 50 cases here. But I agree with Stan. I think narrowing this to just a high-risk recommendation or just a college recommendation will cover just 15 of roughly the 50 cases that are occurring. So I think if we really want to prevent a disease that we can prevent, then we need to make a broader recommendation. Thanks.
I really appreciate the last 3 comments from Dr. Plotkin, Dr. Baker, and Dr. Offit. And just reflecting back on slide number 10 from Ms. McNeil's presentation, the rate in college students was 0.1 case per 100,000, or 0.01 case per 100,000, and 3 times that for the cohort age 18 through 23. Are there further comments? If none, I think we move on to the typhoid vaccines. And if we can have Dr. Lunge come up, or actually Dr. McMahon, come on up and introduce the topic.
Good morning, everyone. So we'll be talking about typhoid and typhoid vaccines now. The current ACIP recommendation for typhoid vaccination was published almost 20 years ago in December of 1994. And so the objective has been to update this recommendation because it is so long out of date. An ACIP work group on typhoid vaccines was not convened because the updated statement doesn't include substantive changes to the recommendations. In 1994, 3 vaccines were included in the statement. They were the live attenuated TY21A vaccine, the subunit, a parenterally administered VICPS vaccine, and a heat phenol inactivated vaccine. The first 2 are still marketed and available in the United States. The third, the heat phenol inactivated vaccine, was discontinued in 2000. And one of the major changes in the update is the removal of that vaccine that's no longer available in this country. In the 1994 recommendation, the statement that either oral TY21A or parenteral VICPS is preferable was made. This is because of the highly reactogenic nature of the heat phenol inactivated vaccine. The vaccination is indicated for 3 groups. Travelers to endemic countries, which continue to be the major group that receives vaccine, household contacts of chronic typhoid carriers, and laboratory workers who work frequently with Salmonella serotype typhi cultures or specimens. These groups have not been changed. In today's session, Dr. Brendan Jackson is going to present information on typhoid fever and typhoid vaccines, a summary of the proposed updates to the typhoid vaccination statement, and then there'll be time for discussion and a vote. So in summary, the 1994 ACIP statement is outdated. In the update, we're proposing no substantive changes in recommendations. The updated statement reflects changes in vaccine availability with the removal of the wholesale inactivated vaccine that's no longer available, newer data on typhoid epidemiology, and newer data on vaccine efficacy and effectiveness, as well as newer data on vaccine safety. So I will turn it over to Dr. Jackson now.
Good morning. So I'll provide some brief background on typhoid fever, since many U.S. clinicians rarely, if ever, encounter the disease. Typhoid fever is caused by Salmonella enterica, serotype typhi, and as opposed to most other Salmonella serotypes, humans are the only reservoir for serotype typhi. Infection is usually acquired through contaminated food or water. The incubation period ranges from 6 to 30 days, and illness has an insidious onset. Typical symptoms include increasing fever, malaise, headache, and anorexia. Typhoid fever can result in life-threatening complications, including septic shock and intestinal hemorrhage and perforation. Typhoid fever can be treated with one of several antimicrobial agents: a fluoroquinolone, a beta-lactam, azithromycin, chloramphenicol, and trimethoprim sulfamethoxazole. However, antibiotic resistance is common and increasing in many areas. The case-fatality ratio for untreated disease was reported at 10 to 20%. However, less than 1%, the current case-fatality rate is less than 1% with early and appropriate antimicrobial treatment. Enteric fever is the broader term for both typhoid fever and paratyphoid fever. And paratyphoid fever is caused by 3 non-typhi Salmonella serotypes, and that's paratyphi A, B, and C. The most common cause of paratyphoid fever is serotype paratyphi A, and this causes disease that is clinically indistinguishable from typhoid fever. It's important to note that current typhoid vaccines provide little or no protection against paratyphoid fever. Globally, there are an estimated 20 million cases of typhoid fever annually, resulting in about 200,000 deaths per year. In comparison, there are an estimated 5 million cases of paratyphoid fever each year. However, in some Asian countries, paratyphoid fever caused by serotype paratyphi A was responsible for a growing proportion of enteric fever cases, accounting for half of enteric fever cases in some areas. Multi-drug resistance, defined as resistance to ampicillin, chloramphenicol, and trimethoprim sulfamethoxazole, is common for serotype typhi. Additionally, fluoroquinolone resistance and extended-spectrum beta-lactamase-producing strains are emerging problems. Growing rates of drug resistance increases the importance of vaccination as a preventive measure. Now, in the US, there were about 400 cases of typhoid fever reported each year from 2007 to 2011, and 90% of these were travel-associated. Among these travel-associated cases, 80 to 85% involved travel to 3 countries: Bangladesh, India, and Pakistan. Use of typhoid vaccines is thought to be low among travelers in general, with one study finding that about 20% of travelers had received vaccine. The most up-to-date vaccine recommendations for travel can be found at cdc.gov/travel. Currently, typhoid vaccine is recommended for travel to most countries in Africa, Asia, and Latin America. Since 2011, typhoid vaccine is no longer recommended for travel to certain countries in Eastern Europe and the Middle East. I'll now provide some background on the typhoid vaccines. So as you've heard, there's 2 typhoid vaccines currently available in the US. There's the TY21A vaccine, which is a live attenuated vaccine administered as oral capsules on 4 alternate days over 1 week. The VI capsular polysaccharide vaccine is given as a single parenteral dose. The TY21A vaccine is approved for persons aged 6 years and older, and the VI polysaccharide vaccine approved for patients 2 years and older. If continued exposure is expected, repeat dosing with the TY21A vaccine is recommended every 5 years currently, and the VI polysaccharide vaccine should be re-administered every 2 years if continued exposure is expected. Note that no booster effect is observed for either vaccine. Conjugate polysaccharide vaccines are available in a few countries. However, a conjugate vaccine is not licensed in the United States. Now, both available vaccines have moderate efficacy based on studies conducted in endemic countries. In a recent systematic review and meta-analysis, the TY21A vaccine had a 2.5 to 3 year efficacy of 48%, but that was based on a single trial. In 2 excluded trials that did not adjust for the cluster design and may have overestimated the protective effect, efficacy was 79% at 5 years and 62% at 7 years. In the same systematic review and meta-analysis, the efficacy of the VI polysaccharide vaccine was 69% at year 1 and 59% at year 2. The cumulative 2.5 to 3 year efficacy was 55%, again here based on a single study. No efficacy studies have been conducted among US travelers, but a recent study found 80% effectiveness of typhoid vaccination among this group. Note that this estimate is for any typhoid vaccination, as the study was not able to differentiate between the 2 vaccines. In terms of safety, both vaccines are generally well tolerated with low rates of adverse events based on data from trials and post-marketing studies. In the meta-analysis of field trials, 2 events were more common in vaccinees than among placebo recipients, and this was fever with a risk ratio of 1.8 and a combined measure of any mild adverse event with a risk ratio of 1.7. In an analysis of post-marketing data from the Vaccine Adverse Events Reporting System, there were an estimated 0.6 serious events reported per 100,000 doses distributed. Serious adverse events were defined as reports of death, hospitalization, prolongation of hospitalization, permanent disability, life-threatening illness, or congenital anomaly. For the VI polysaccharide vaccine, the meta-analysis found that pain and swelling at the injection site were much more common among vaccinees than among placebo recipients, with risk ratio point estimates of 8 and 6, although noting the lower bounds of the confidence intervals. In the VAERS post-marketing study, the VI polysaccharide vaccine was also associated with a similarly low rate of serious events. Contraindications for both vaccines include hypersensitivity to any components of the vaccine, and then ACIP's general recommendations on immunization suggest that live bacterial vaccines like TY21A are generally contraindicated in pregnant women. Additionally, the TY21A vaccine is contraindicated in immunocompromised persons and should not be administered during an acute febrile illness. Because the TY21A vaccine contains live bacteria, persons should avoid taking antimicrobial agents if possible 3 days before and after administration of the vaccine. However, certain antimalarial prophylaxis medications can be taken at the same time as vaccine, which is relevant given many patients are travelers. Also, TY21A can be co-administered with other live vaccines, including the yellow fever vaccine. The VI polysaccharide vaccine should be given to pregnant women only if clearly needed. So in these following slides, I'll show the minor proposed updates to the current ACIP recommendations for who should receive vaccine. So the first group for whom vaccine is indicated, as you've heard, are travelers to areas where there is a recognized risk of exposure to Salmonella serotype typhi, and there's really no substantive changes to this one. And readers are directed to the current CDC website for most updated guidance on countries. So this recommendation goes on to include information to state that the risk is greatest for travelers to developing countries, including those in Latin America, Asia, and Africa, and who have, especially for those who have prolonged exposure to possibly contaminated food and drink. However, we did add that short-term travelers are also at risk based on a recent study. The text notes that multi-drug resistant strains of Salmonella serotype typhi have become common in many regions, and that cases of typhoid fever are treated with drugs to which the organism is resistant can be fatal. Finally, the recommendation reminds clinicians that travelers should be cautioned that typhoid vaccine is not a substitute for careful selection of food and drink, since typhoid vaccines are not 100% effective, and that vaccine-induced protection can be overwhelmed by large inocula of Salmonella serotype typhi. Again, no substantive changes here. So it's also, typhoid vaccine is also indicated for persons with intimate exposure, for example, household contact to a documented typhi chronic carrier. And a chronic carrier is defined as excretion of typhi in urine or stool for greater than 1 year. So the change here to the 1994 guidelines are that carriers are further specified using the word chronic, and that chronic carriage is explicitly defined. And the purpose of this change is so that the recommendation is not misconstrued as applying to close contacts of any person with typhoid fever and not just chronic carriers. The final group for whom vaccine is recommended are microbiologists and laboratory workers who work with cultures of Salmonella serotype typhi or with specimens that contain this organism, or who work in laboratory environments where these cultures or specimens are handled. This text was suggested by laboratory colleagues to clarify these 1994 recommendation, which was microbiology laboratorians who worked frequently with S. typhi, with the nomenclature change of typhi from species to a serotype there. So regarding choice of vaccine, we propose no substantive changes. As in the 1994 statement, no preference is given between the parenteral VI polysaccharide and oral TY21A vaccines. Both are acceptable forms of immunization. In choosing a vaccine, clinicians should consider approved ages for use, dosing, and contraindications and precautions. The inactivated whole cell vaccine that is no longer available has been deleted from the recommendation, as you've heard. So again, in summary, the current 1994 typhoid vaccine statement is outdated. No substantive changes and recommendations are proposed. And the updated statement is presented today for ACIP vote. It reflects a change in vaccine availability, with the whole cell vaccine being removed, new data on typhoid epidemiology, and newer data on vaccine efficacy, effectiveness, and safety. Thank you.
I'd like to thank the two presenters for a very succinct presentation. I just want to make the reminder that ACIP strives to, on an every 5-year basis, review and renew, reaffirm, retire, or revise its statements. This is now a 20-year-old statement. And again, the biggest change has been the loss of one of the vaccines and some minor wording changes. We did have a little bit of a discussion, as Dr. Jackson knows, in terms of whether or not this should be subjected to a formal grade review. And a couple things. One, there has been kind of a shortage of excess time around CDC over the last few months, or so rumor has it. The other thing is that really nothing has changed from the original. There have been systematic reviews that basically suggest strongly that there is efficacy as previously stated, and the safety has not changed at all. So I just want to make those comments. Dr. Schaffner had his hand up.
Yeah, thank you, Bill Schaffner. I note that 90% of the cases are travel associated, and of those, 80 to 85% are travel to Bangladesh, India, or Pakistan. I was curious as to who these travelers are. I'm thinking analogous to malaria. Are these people who were born in the United States and are traveling to those countries, or are these, many of them, people who were born in those countries, have come to the United States to live, and have now gone back to be with family and visit? This is very important because if the latter, perhaps a sentence or two might emphasize that those are also individuals at risk, because we see those folks often not taking malaria chemoprophylaxis when they go back to their home of origin, their country of origin.
So I'm not actually familiar offhand with the statistics on those that are visiting friends and relatives originally from there. Maybe Dr. Mann has information on that.
Yeah, most of the travelers are people who are visiting friends and relatives, not necessarily who were born in those countries, but visiting friends and relatives.
So perhaps a little bit of a sentence or two to include those folks, or highlight the fact that those folks also are at risk.
Thank you.
Dr. Messonnier.
Hi. In your attempt to clarify the laboratory workers, you actually confused me. So are you talking about laboratory researchers who work with this organism, or are you including a local clinical laboratory where they may indeed get a patient returning traveler and therefore come through their laboratories a specimen? And have you quantitated how many people would be in this category? If you do mean the local laboratorians who might get in, contact the organism, it's actually quite a large group of people who you're recommending for.
Right, and we were just revisiting that recently, and I think it is a concern having that broadened recommendation that it may apply to more people than initially intended. There are cases that do continue to occur among laboratory workers and can be among clinical laboratory workers, but they are few and far between. So one thing we might consider doing is adding the word frequently, work frequently with Salmonella serotype typhi. And we could consider revising that in general about the specimens, the handling of specimens.
Dr. Freithofer.
A quick question. Does the oral vaccine still need to be refrigerated?
Yes, it does. Thank you.
And Dr. Pickering.
Yeah, to follow up on Dr. Schaffner's question, this is not a group that need to be immunized, but we often see children who are exposed to people who have traveled to these areas of the world and come back are themselves asymptomatic, but the children who are exposed to those individuals who have traveled develop typhoid fever.
Dr. Bologna.
Another follow-up on Dr. Schaffner's question, and that's conversely, there seems to be relatively a small proportion of cases coming from Latin America. And so my question is, has the epidemiology changed in Latin America? Has the risk decreased there?
Yeah, thanks for the question. I don't recall those precise percentages, but we do see cases, travel-associated cases from Latin America. I think I've read that overall the incidence has been decreasing in Latin America, but we definitely see cases. We also see quite a number of cases from the Caribbean. So those areas are still recommended. Travelers to those areas are recommended to receive vaccine.
Dr. Smith.
Hi, thank you. This is Jean Smith, CDC. I have a question. I don't know if you can answer it, Barbara or Brendan, but I was struck by only 20% of travelers receiving vaccine. My daughter went to India and Nepal last summer, so I paid for the oral typhoid series at Publix. I think it was about $50. So do you think, is this a cost issue or lack of awareness, or maybe you don't have that information?
We really don't have that information. We'd like to get it. But I think that the issue that Dr. Schaffner was bringing up, I suspect is really central to the low rates of vaccination, which is that many of the travelers who are at risk for exposure are people who are visiting friends and relatives and who simply don't think about it. It just doesn't, it feels like going home and they're not really thinking about vaccination for travel. So I think that efforts to reach those groups would really be the most effective way of decreasing rates of typhoid in this country.
So you mean that they're like, for example, non-resident Indians? And how do you have that information?
So we know whether people are traveling to visit friends and relatives because that's reported as part of the enhanced surveillance for typhoid in this country. I can give you just an anecdote from a colleague who is of Indian descent who we were discussing this session with earlier this week, and he said he kind of got a startled look in his eye and said, my father's visiting India right now, and it never occurred to me to tell him to get vaccinated.
Dr. Lehr.
Just clarifying, because the oral vaccine is a live attenuated vaccine, it probably has the same rules of 28 days away from other live attenuated vaccines such as measles and varicella. Is that true? And if it is, you might want to point that out in the statement.
Sure, and I'll definitely review the information again, but as I recall from the 2011 general recommendations on immunization from ACIP, there was, I believe the language said there was not a contraindication to administering these live vaccines together with typhoid. And anyone can correct me if I have that wrong.
Correct. It's simultaneously is fine, but if you give one, then you need 28 days between another live attenuated.
Again, thanks for the comment, and we'll need to go back and check. But I think because it's orally administered, I think that may be the difference. But with the parentally administered live viral vaccines, the biology is different.
And Ms. Pellegrini.
Thank you. I was puzzling just a little bit over the precaution that the vaccine should only be given to pregnant women if clearly needed. And I was trying to think about some cases where it might be indicated but not clearly needed. Are you thinking, for instance, you're going maybe on a cruise and it has a port or two in Central America, but you're not going to have that prolonged contact? Is that the sort of circumstance where maybe a pregnant woman shouldn't be vaccinated?
That statement actually comes directly from the current vaccine label, so there was no really change from that. So we definitely could discuss that more, but I don't think we've actually given exact thought to how that should be interpreted. It's just basically based on the FDA-approved label.
And I do think, though, that the kind of situation that you're bringing up would be exactly the sort of situation that this would apply to. So, someone who is clearly going to be in a setting with very limited access to clean food and water for a prolonged period of time during pregnancy, the risk-benefit balance would be shifted from someone like the hypothetical person you just raised.
I believe Dr. Kroger might have an answer for Dr. Lair in terms of spacing.
Yes, with respect to the 28-day rule for non-simultaneous vaccination of live vaccines, the General Rex applies that to injectable vaccines and intranasal vaccines, but not oral vaccines, so it would not apply in this case.
Thank you for that clarification. Dr. Decker.
And with respect to the question about the pregnancy language, that's regulatory language that's prescribed by the Code of Federal Regulations. So you need to interpret it with that in mind.
Thank you. Other comment? As this is a recommendation for a vote, and notwithstanding the changes that we have suggested to better describe the frequency of exposure for microbiologists, some nuances in terms of pregnancy, including the language in terms of the spacing not being an issue, in terms of a live oral bacterial vaccine. And there was one other that is escaping me at this point in time. But if that's the case with these minor changes we've mentioned, do we have a motion for approval? Dr. Reingold. And do we have a second? Dr. Rubin. Further discussion? Wonderful. Dr. Bocchini. And to your left, we will move to Dr. Riley. So Dr. Bocchini?
Okay. Bocchini, yes.
Laura Riley, yes.
Allison Kemp, yes.
Karen, yes.
Belongia, yes.
Harrison, yes. Romero, yes.
Rubin, yes.
Bennett, yes.
Tempte, yes.
Pellegrini, yes.
Reingold, yes.
Vasquez, yes.
And the motion passes 13-4, 0 against, and 2 absent from voting. And we thank I can't say the work group for their efforts, but I do thank Dr. McMahon and Dr. Jackson for putting this together. And let's have Dr. Shimabukuro up to the microphone on vaccine safety and changes or proposed changes to the VAERS reporting form.
Good morning. Today I'm going to give you an overview of the Vaccine Adverse Event Reporting System Form version 2.0 proposed. I'll give you some brief background on VAERS, go over the VAERS form, and then activities and next steps. So VAERS is the National Spontaneous Reporting System for Adverse Events after U.S. licensed vaccines. And in recent years, we've received about 30,000 U.S. reports annually. VAERS accepts reports from anyone. Signs and symptoms of the adverse event are coded using medical dictionary for regulatory activities terms. VAERS is jointly administered by CDC and FDA, and it was authorized by the National Childhood Vaccine Injury Act of 1986. So as far as submitting a VAERS report, currently there's a secure online submission process, and that's roughly 30% of reports in recent years. Most reports come in as mailed hard copies or faxed hard copies of the paper form, and in rare instances, via telephone through a VAERS customer service representative if there's no other way to submit a report. The rough breakdown of reporter types is about a third are vaccine providers, about a third are vaccine manufacturers, and about a third are patients, parents, or other, although there's variability among individual vaccines. So this is a miniaturized version of the VAERS-1 report form, the current VAERS form. It's a paper form. It's been in use since 1990. It must be completed by hand. You have to download it, print it, complete it by hand. Forms are mailed or faxed to the VAERS contractor, and it requires manual processing and data entry procedures, which are fairly resource intensive and time consuming. Hard copies are also scanned and uploaded into the VAERS image database. So there's a database of a traditional research type database, and then there's an image database of the actual report. I just want to show you this. These are a couple screenshots of the VAERS online reporting tool. We prefer that VAERS reports get submitted online. As I said, it's about 30% of reports. And even after we implement the new electronic form, we would still prefer that VAERS reports come in through the online reporting tool. It has the same fields as the VAERS-1 form, but in a different presentation. The disadvantage of the online reporting tool, which is a barrier for some types of reporters, is that the interface will time out. So for security reasons, you must complete the report in a single setting. If you leave the report for a certain period of time, it'll time out and you'll lose the report. So the objectives for the VAERS 2.0 form proposed was to create a fillable, savable electronic reporting form, update data fields to address current vaccine safety information needs and changes in vaccine practices over time, modernize the appearance and format of the VAERS form, modernize reporting procedures, specifically to implement electronic document upload capability, and I'll discuss that a little bit further. Ensure data collected on the VAERS 2.0 form allows for comparisons to be made with older data. So we want to be able to make historical comparisons between VAERS-1 and VAERS 2.0 data. So why are we doing this revision of the VAERS form? Well, some fields on the current VAERS form have limited public health and or regulatory value, and we determined that by through meeting with our own ISO staff that work with VAERS data on a daily basis and our colleagues at FDA that use VAERS data for regulatory purposes. And on the flip side of that is other information, important information isn't being collected. Some fields are no longer relevant due to changes in the immunization program. The language in some fields is confusing and needs clarification. Fields used in paper reporting or for manual processing will no longer be necessary. So certain fields like that the manufacturers use or that the VAERS contractor used to manually process reports will no longer be necessary, and the manufacturers are moving to a different reporting process. And also new fields such as pregnancy are needed because of new recommendations and patterns of vaccine uptake, and we heard this from NVAC and other stakeholders that made recommendations for some new fields. So continuing on, so why revise the VAERS form? Well, handwritten, mailed, and fax paper reporting is an inefficient way to conduct vaccine safety surveillance. And paperless reporting using an electronic form would eliminate most manual processing and much data entry. It would mitigate problems with poor handwriting and non-standard reporting, take advantage of smart features such as auto-population, where, for example, if we have a valid date of birth and a valid date of vaccination, we can auto-populate the age of the patient. You can incorporate drop-down menus, program checkboxes, pop-up instructions, and logic checks, which are important because we can address the issue of illogical answers like the adverse event onset date being before the vaccine was administered or before the date of birth. It'll allow for standardization of data elements, and it'll also address the complaint of getting timed out, like I mentioned. The VAERS form, since it's an electronic fillable, savable form, can be completed in stages, and multiple people can work on it, and it avoids the problem of losing an electronic report if you can't complete it in a single setting. And then manufacturers are transitioning to fully electronic reporting using the ICH E2BR3 message standard. And so it made sense to transition non-manufacturer reporting to electronic. So actions that have already occurred, there's been initial internal development, review, and revision by CDC, FDA, and the VAERS contractor. And I'll say revision is ongoing. There's external review by immunization partners at CDC, within the federal government and outside the federal government. Cognitive interviews with potential reporters to include physicians, nurses, pharmacists, parents, and patients. And revisions based on the results of cognitive interviews. And I'll just mention that cognitive interviews are commonly used to validate questions in surveys. We presented to the Federal Immunization Safety Task Force, conducted follow-up interviews with a sample of individuals that completed the initial cognitive interviews. We presented to the Advisory Commission on Childhood Vaccines and to the National Vaccine Advisory Committee. Our contractor conducted computer testing of the SMART form with potential reporters, and we made revisions based on the results of the computer testing. And we recently presented at the FDA Electronic Post-Market Safety Reporting Updates meeting. So our proposed reporting method would be that for this electronic form, the reporter would download the VAERS form from the VAERS website. Currently, you have to download the VAERS-1 form in order to print it. So downloading the form from the website, completing the form on a computer, saving the VAERS 2.0 report as an electronic document in a local environment, and then uploading the report to the VAERS contractor through the VAERS website. The contractor then electronically extracts the data from the VAERS 2.0 report into the VAERS data, thereby bypassing much of that manual data entry and manual processing, and would also do some reviewing, redaction, and QA on the data. And then the VAERS contractor would generate an individual report for the VAERS image database. So we wouldn't be scanning reports and uploading them into the image database. We would just create a report based on the extracted data and post that on the image database. And I will say, when we do implement reporting, we'll still have options for manual reporting like faxing and mailing, at least in the short term, maybe until some of the technology and connectivity catches up. So this is a miniaturized version of the current VAERS form, which is available online at that link. And then this is the VAERS 2.0 form proposed, and you have a full-scale version of this in your binder, or there were handouts for this particular presentation. So the changes to the VAERS form can really be summarized by three categories. There's tweaks, there's revisions, and then there's new questions. So tweaks are really clarifications or saying things more plain language, or just minor additions to certain questions or data elements. Some of the data elements stayed the same, so there really was no change. A revision would be a change in the language to get at the same information. So an example of a revision, on the VAERS-1 form, there is a question that says responsible physician. And when we cognitively tested that question, a lot of the physicians felt that they had a problem with the way that was answered, because responsible physician to them implied responsibility for the event or the outcome, possible liability. They recommended we made some changes to that. So our change was to change the question responsible physician to best doctor or healthcare professional to contact about the patient. That's just an example of a revision. So I would say the overwhelming majority of changes to the VAERS form are either tweaks or revisions. There are several completely new questions or new data elements. There is a question about pregnancy status, and there are questions about race and ethnicity, and there's several other new questions that that if you look at the VAERS form and compare it side to side, the 2.0 with a -1, you can see where there's new questions. So next steps, present to ACIP, which we're doing today. Public comment solicitation through the Federal Register. We have one minor change to make on the current draft of the form, and that'll get posted on the Federal Register for 60 days. We'll then make final revisions. We'll then develop the platform to accept the electronic VAERS 2.0 submission and update the online reporting tool to reflect the new data elements. So once we finalize the new questions, we'll go and we'll update the online reporting tool with the new questions. Then implement the VAERS 2.0 form, then evaluate the completeness and quality of VAERS data, really a pre-post comparison. So that's my presentation. I'll be happy to answer any questions.
Thank you very much, Tom. I have two really quick questions. First, will there be any forced answers on the new form?
No, there's no forced answers. So there will be pop-up reminders. If a person puts in an illogical answer, there will be a pop-up reminder cautioning them that answer may be wrong or they may want to reconsider. I don't know what the exact language is. However, the person always has the option to override that or leave anything blank that they want to. If there are answers left blank, there'll be reminders saying you left these fields blank, these are critical fields, but you never get a hard stop on the form.
Thank you. The other question is, it sounds like this is going to be going to an external contractor for extraction, and then the question is whether or not there's going to be any verification, of the incoming information, and then what do you have after it comes back from the contractor?
Well, currently the VAERS report intake, processing, and data entry is done by a contractor. So there's really no difference in the way things are happening now, except instead of the paper reports getting mailed or faxed to the contractor, we hope that most of these forms will get electronically uploaded to the contractor.
So the likelihood is actually less variability then, going from the forms to the final data?
We would hope so, yes.
Okay, thanks. Other question? Dr. Rowley.
Thank you for now including pregnancy-specific information in this, but just as one plea, it says under 8, if you're pregnant, yes, and then it says in the parentheses, that's where you put sort of the pregnancy information. It goes into box 18. The biggest problem with the form and sort of then trying to figure out what to do with the information is that we almost never know either the due date or the gestational age, and so you can't time when the vaccination was given, right? So I think you have to force people to put due date down here. You mentioned that it should be done, and then in 18, and then on the back under item 18, it never comes up again to put the pregnancy information in there. It's such an important critical element that I feel like it needs to be sort of bolded somehow, because it makes pregnancy information from this useless. Because we keep trying to draw conclusions about vaccines and pregnancy, and the vaccines that are given in the 3rd trimester didn't cause a birth defect. So until we get the due date or the gestational age, we're stuck. We're always going to have sort of this bad data that no one knows what to do with.
So in previous iterations of the form, we had much more detail on pregnancy, and we actually, I think 3 of the physicians we did interviews with were OBGYNs. Yeah. The feedback we got was not just with pregnancy, but with the form in general, was simplify it and have people fill in information in the text box. And we thought that by putting check boxes in or specific questions for specific information, initially we thought that was a good thing and that would help. But when we got to the cognitive testing, which actually challenged a lot of our assumptions, the feedback we got was direct people to put that information in the text box. So I think, you know, we do.
Suggesting that you want to put in like, you know, birth defect and, you know, miscarriage and preeclampsia. And, you know, they can write that in if they want. But I think the one critical piece of information that you really need to know is EDC, you know, estimated due date or EDD, however you want to call it, because
Do you think
can't draw anything from it otherwise.
So we have some instructions below the question on box 8. I mean, would you suggest putting something in there to highlight, and maybe we can work to figure out what are the most important things and put them in the front of those instructions.
Yeah.
But
either that or on that line where it says, if you checked off pregnancy, yes, then put a little box saying EDD so that you're forced to put in the gestational age. That's it. And the rest of the pregnancy stuff, if you want to write it down, you can put it in the box under 18 where you said to put it in. But it's such a critical element, and that's a common thing that OBs write down all the time. It's like the one box we do fill out, honestly.
Okay, I think we have some of the. I will say, just as a general statement, I know there's probably a lot of people in this room who have seen versions of this and made great suggestions and suggestions that are perfectly reasonable. If you don't see them reflected on here, there's probably 2 reasons why. Number one, just a pure space consideration, and number 2, it may not have tested well when we actually went out and tested it. But I think there's probably enough space in that to work with you to address that concern.
Dr. Lehr?
So 2 suggestions there. One would be in 18, just have a simple question, if pregnant, due date. And on the electronic version, you could say if 8 is marked yes, when you get to 18, you simply have to, you give them another question under 18, if pregnant, due date. So it's just a positive only if they've marked number 8 yes.
Dr. Belenja.
And just to kind of follow up on that, you know, as I understand it, this is a real nice improvement. I think it's great. It's essentially, though, you're still filling out a form completely. It's just that it's going to be online instead of a hard copy. And maybe for version 3, when you get to that, you know, you can take advantage of the computer technology that allows you to have branching points. And so not everybody has to see the entire form. They see whatever is relevant for whatever they're filling out. And so there could be a different pathway for someone who's pregnant where you collect other information, but other folks don't even see those variables because it goes down a different path.
I think for the online reporting tool, that may be an option. So I talked about like the smart form. It's actually not that. It's not all that smart. Like an online, the online version, we can build a lot of things into it. But the PDF smart form is actually limited in its smartness. So.
Comment up at the microphone.
Yeah, thank you, Jane Zucker from the New York City Immunization Program. So we happen to have a fairly robust registry, and what we do now is we actually have an adverse event reporting module that takes the information from the vaccine event that was reported, so it automatically pulls out the lot number, you know, vaccine information, and we generate a VAERS form and fax it. So I think having the electronic online system will be great because we'll transition to actually be able to electronically submit it. Again, pulling all of the information, demographics that we have on a child, all the vaccines, lot numbers, et cetera, that they received that day, to pull it and send it to VAERS. So it's just more a comment about the stakeholders that you mentioned, and not to forget about the immunization information system folks and the program managers, because we're the ones that will actually take the information and the specifications you give us from the online form and build that into our registries to try to improve reporting of adverse events as well as the quality of the data that you're getting. So thank you.
And one more comment.
Carol Hayes with the American College of Nurse Midwives. We would wholeheartedly and completely support Dr. Riley's statement that without an actual estimated date of delivery, your data is absolutely useless. So please add that. Thank you.
And I assume that pertains only to pregnant patients. Thank you very much.
I will say one problem with putting the pregnancy into the box 18 is you'd be amazed what people will write if you tip them off to write about something. So I think we used to have much more descriptive language in box 18, and in the cognitive interviews, people were a lot of physicians were saying just say describe the event and leave it to us to just report it in there. So sometimes you can, from our research doing this, you can sometimes the more detail you ask, the more problems you potentially create or unintended consequences.
Dr. DeStefano.
Yeah, I just want to make the comment that our data gathering doesn't stop when we receive the forms, and in particular for pregnancy, that's been a high priority for our office, obviously, for a few years now, and that means when we get reports of pregnancy, we have gone on to get additional detailed information about the pregnancy, the delivery, et cetera. So we do get the information about birth outcomes, the date of delivery, expected date of conception, and such. We do get those if they're deemed important in follow-up data gathering.
Thank you for that comment. Other comments? Seeing none, we have a break. You guys have done wonderfully for a 3-hour stretch here. We have a 10-minute break. We'll resume at about 10 after 11.